Welcome to your human variation and genetics lab. And notice that I don't have any PPE on here, and you don't have to either, because this is a dry lab. And in fact, the only material you're going to need for this lab is a penny and maybe some colored pencils, and that's it. So I have the lab handout here so that you'll be able to see it. And I have something else here. I have a nice partner, and you're going to need a partner for this lab because we're going to be parents making an ugly baby. So you're going to need a mom and you're going to need a dad. Now, it doesn't have to be a partner of the opposite sex. It could even be your pet dog. Doesn't really matter. But in this case, my lab partner who's going to pretend to be my mate happens to actually be my mate. This is my husband, Paul. And we really have a real beautiful baby in real life. But today we're going to be making a fake ugly baby. All right, so basically what this lab is going to do, it's going to allow you to do a hands-on part first that will help you practice basic classical genetics, Mendelian genetics. Specifically, you're going to be practicing Mendel's law of independent assortment, which says that during meiosis, when homologous chromosomes pair up down the middle, you could have mom's chromosome for a particular set on one side and dad's on the other, or by the law of independent assortment, they could assort the opposite way. And that's true of each pair of chromosomes. For humans, that's 23 pairs of chromosomes, ensuring genetic diversity of the baby that could be formed as a result. Now, we're also going to practice the law of segregation. And Mendel's law of segregation, and again, Mendel was a genius because he figured this out long before DNA was ever discovered, but he figured out that during meiosis, these genes are going to assort independently and sort separately into separate gametes, sperm for males, eggs for females. So in other words, each sex cell gets just one copy of each trait. In other words, you get one allele for hitchhikers in this sperm and one allele for hitchhikers in that sperm. You never have two traveling together unless you have a genetic abnormality resulting from non-disjunction. So all of that stuff was reviewed in lecture, so go back and learn it a little more. Now, one of the things you're going to want to really make sure you do before you begin this lab is make sure you understand the definitions of all these terms here. Learning genetics is very heavy on vocabulary, and if you don't understand the vocabulary, you're going to be kind of lost. So if you need to, go back and study these vocabulary words here. Now, the first part of the lab, as I said, is hands-on with this penny. The second part is just practicing genetics problems. So that part's a little less ambiguous. So here's what you're going to need to do. Grab your partner. Again, I got Paul here. And we're going to be a mom and a dad. I'm going to be mom. He's going to be dad, but could have been the other way around, however you want to work it. And we're going to be looking at a whole lot of different traits that are going to make up our baby. Specifically, you're going to be looking at 29 different traits, all the way from whether the baby gets bushy eyebrows to whether they have a cleft chin. Now, we're going to practice with just a simple example that won't be officially part of the lab, and that's hitchhiker's thumb. Now, for each one of these 29 different traits, we're going to assume that each of us has a heterozygous genotype for that trait. And you'll recall heterozygous means each of us carries one dominant allele and one recessive allele for that trait. So, for example, if we were looking at hitchhiker's thumb, notice Paul in real life does not have hitchhiker's thumb. He has straight thumb, but we would assume that he is heterozygous for hitchhiker's thumb, big H, little h. So how are we going to determine which allele gets passed to this baby for each trait? Well, we're going to use this penny to illustrate the effect of the law of independent assortment. So each of us, for each trait, will flip the coin. And you want to do it like this so that you have the best chance of getting 50-50. Just flip, grab, put it over. And in this case, I got heads. So heads is going to represent that I donated a dominant allele to that particular egg that got fertilized and became the baby. Whereas if I got tails, it would represent that I donated my recessive allele. Because remember, I'm heterozygous, big H, little h in the case of hitchhiker's thumb. So because I got heads, I donated that big H into the sex cell that became the baby. Now, before we get started, let's, figure out what we're going to name our baby. And actually, you might want to figure out what its sex is first. So you're going to want to have the father flip to determine the sex of the child, because that's what happens in real life. Remember, when you're talking about sex chromosomes, and that determines whether you have a boy or a girl, Females, normal females, are XX. So we can only make eggs that carry the X chromosome. But males can make two types of sex chromosomes, X or Y. So he needs to flip to determine, is he going to pass an X chromosome onto his baby-to-be or a Y chromosome onto his baby-to-be. So Paul, go ahead and flip for that. That was a terrible flip.
It was good enough.
He got tails. So because he got tails, that means we're going to assume he donated an X to this baby. So he donated an X-bearing sperm. And of course, I could only donate an X, so our baby ends up XX, and it's going to be a girl. Okay, so keep that in mind. What are we going to name our little girl?
I haven't decided yet.
Flippa?
Because if it was a boy, it'd be Flip, right?
Right. Okay, our girl's her name's gonna be Flippa. Okay, so it says to record it in the chart. So now we're gonna scroll down here. And this is the data chart that you're gonna be filling out. So notice I put in my name Bree here. The other parent's name is Paul. And because this is a girl, it's gonna be Flippa. And because he donated an X instead of a Y, I donated this first X. He donated a second X, so instead of a boy, this is going to be a girl. Okay, so this is a girl, and her name is Flippa. It should be two P's, I think, right? Flippa?
I don't know.
Okay.
We flipped a coin.
We flipped
That's
a coin,
where that
so it's
comes
Flippa. Okay, so the first set I've given you is an example. You actually won't be needing to do hitchhiker's thumb, but we're giving this as an example. So each parent is going to need to now flip the coin to determine which allele we donate to the baby. Now remember, each of us, we're assuming, is heterozygous for each trait. So each of us in this case would be big H, little h. So if I'm big H, little h, I can donate either a big H to my egg or a little h to my egg, but I need to flip to determine what it's going to be. So we're practicing the law of segregation and independent assortment here. So flip, boop. I got heads. That means I donate a dominant allele. So heads represents a dominant allele, tails represents a recessive allele. So I'm going to change this to big H. Daddy, you flip now?
That was a bad flip, but there it is anyway.
Okay, he got tails, which means he donates a recessive allele. So we're going to represent that as little h. So because I had a big H and he had the little h, we do the hokey pokey and the baby comes out heterozygous, big H, little h. And you might recall hitchhiker's thumb is a recessive trait, meaning to get hitchhiker's thumb, I would have needed to have received a recessive allele from each of my parents and been homozygous recessive, little h, little h. But in this baby's case, I donated a big H egg and he donated a little h sperm, so this baby ends up with a straight thumb, not with a hitchhiker's thumb. Okay, so that's just one example. You're going to be doing a whole lot of traits. And so included in this handout is a little sort of booklet, appendix, that goes through all these different traits you see here. And a few of them don't follow simple Mendelian genetics. They are unusual non-Mendelian cases. So I'll get into those in a sec. I've highlighted some of those. But let's start with face shape. I'll just do a few of these and let you guys carry on from there. So this face shape, now if I scroll down, you'll see that, that this little guide to facial traits goes in over a whole bunch of these. So face shape, it could be, I could have a baby that has a round face, and it turns out this is a dominant trait. So to get round face, I'd have to be homozygous dominant, big R, or heterozygous, big R, little r. If I come out homozygous recessive, little r, then I inherit a square face. So let's scoot back up here to the chart. So again, we're assuming each parent is heterozygous, so in this case, big R, little r. And remember that the two alleles separate independently and into separate gametes during meiosis. So allele from the mom, am I going to donate a big R or a little r? Flip, flip. It's heads, so I donated a dominant allele. So I'm going to put in this case, big R. Okay, and Daddy, go ahead. He got tails again, so he donated a little r to his sperm.
My coin flip hit the computer.
That was a horrible coin flip. He just gave me a lecture on flipping coins, and now he's doing a terrible job of it. Okay, so we know that even though my genotype was big R, little r, I donated the dominant allele to the baby. He was big R, little r, but he donated the recessive allele to the baby. So now we can figure out the baby's genotype. And what would the baby's genotype be in this case?
If I can remember, it was round face.
Nope, that's the phenotype, right? So remember
I'm
that.
sorry, I wasn't listening. The genotype, I'm sorry, is heterozygous.
Right.
Big R, little r.
Right.
I jumped
okay,
ahead.
so when the mom's allele combines with the dad's allele at fertilization, this baby ends up with a full diploid genotype, so it'll be big R, little r. Oops. So that's the baby's genotype. In other words, the alleles it inherited from the mother and the father. The phenotype is the expression of the genotype. So in this case, again, if you need to, you can scroll back down to the guide and recall that big R, little r gives us a round face. So scrolling back up, this baby has a round face. And that's how you do that first trait. Okay, so if you wanted, if you had your colored pencils ready and a piece of paper, you can draw a round face, or you could even do this in Word, and I think that's what I'm going to do. Let's go to chin size. Scroll down to chin size. Now notice it says chin traits, next 3 flips. So we're actually going to be looking at 3 different traits for creating a chin. So it turns out chins, and you can see Paul has a very complicated chin, which our real daughter in real life happened to inherit. So now we're going to do chin size. So if you have a very prominent chin, that means you were homozygous dominant, and we're going to call this trait, we're going to use V's for this one, or heterozygous. And if you have a less prominent chin, you're little v. So let's do that because the outcome, in the case of chin shape, it's one of these unusual genes. In order to figure out the chin shape and whether or not it has a cleft chin, is going to depend on the outcome of chin size. So let's go ahead and do chin size, and then we'll get to the second set here. So let's do chin size. We're going to call it V. Use the V, and remember, we're both starting out assuming that we each are heterozygous, big V, little v. So mommy's going to flip. I got heads. Wow, I keep getting heads. So I'm going to donate a capital V here, a dominant allele.
And I keep getting tails.
And daddy keeps getting tails. That's really strange. Okay, so he gets a little v. So once again, the baby ends up heterozygous for this trait. And by coincidence, it just keeps happening like this. So this baby ends up with a prominent chin. And this is very interesting because, again, this is an unusual chin, follows some unusual genetics. So the fact that the baby has a prominent chin is going to influence the next 2 genes, chin shape and cleft chin. And let me see if I can show you real quick why this is. I'm going to open up a new page here just to kind of illustrate this. So it turns out that where genes are located on the chromosome can influence how they're inherited. So I'm going to draw a big X here to illustrate a chromosome. So I'm going to make this guy really big. I'm going to make him like that. Okay, actually, I'm making him bigger than that. I'm going to make him like that. Whoa, okay, that's a good chromosome. And I'm going to put another chromosome here to represent the other parent's chromosome. Now, if I go up to here and I draw, I'm going to draw just a little squiggly line here. And I'm going to make it a different color. I'm going to make it red. Let's say that right here, oops, this is supposed to be a drawing and that didn't work. Let's say right here on the chromosome, that little spot on the chromosome happened to be where we have the gene for chin shape. Okay, so I'm gonna insert a text box here just to show you that this is chin shape right here. Chin shape. Of course, we don't need that to be that big. Okay, so I'm going to do chin shape. And make that guy like this. Okay, so imagine that was chin shape right there. Better draw an arrow just to make that more clear. Okay, so right there is chin shape. Now, there could be, keep in mind, there's a total of 30,000 genes in our genome. And many of these genes are located very close together on a chromosome. So if that was the gene for chin shape, let's say we had another gene. I'm going to insert another drawing. Let's say we had another gene close to it on the chromosome. And I'm going to illustrate this with a different color. Let's see, maybe I'll illustrate this with red. Maybe that gene coded for the next thing, which is chin size. Okay, that was supposed to be an arrow, that didn't work. Insert. And we'll call this chin size. So let me see if I can insert another text box there. Chin size. So that's the second thing that we're doing here. It's smaller. And no fill. And no line. Okay. Okay. So in other words, what I did here was I have two genes located next to each other on a chromosome. One codes for chin shape, the other one codes for chin size. So in this case, and this is the way most genes probably work in the body, whether you or not you get a particular chin size is influenced by what kind of chin shape you got. And the reason why is during crossing over, during metaphase, these guys tend to cross over at the same time because they're what we call linked. They're linked together because they're close together on the chromosome. And so they influence each other. So this is a case of what we call epistasis, that the expression of one trait influences the expression of one or more other traits. So if we go back to our lab handout, we see that that's what's going on here. Let's scroll down to the handout. So notice that for chin shape, and I actually have these guys backwards, it says that in order to inherit the kind of shape of chin you have, it's dependent on what kind of size chin you got. So it says the genotype, it says you only flip the coin for this trait, chin shape, if the chin size you got was homozygous dominant or heterozygous. So remember that in our example, we found that our baby was going to have a prominent chin, so because she was big V, little v. Therefore, we can go ahead and flip for chin shape to see if she has a round chin or square chin. If, on the other hand, we had gotten that the baby was homozygous recessive and had a less prominent chin, therefore that trait does not allow for the expression of this trait because it did not, we did not have that trait travel during crossing over. So, but in our case, we did get the trait for chin size, so we can go ahead and flip for chin shape. And for this one, we're going to be looking at R's again for this. Yeah. So yeah, round chin versus square chin. Okay, so I flip again, and I got heads. Yeah, again. So in this case, it's another big R. So I highlighted this just to remind you that this is a case of epistasis. You know, the expression of chin shape and cleft chin is dependent on what we got for chin size. Wow, he got dominant as well. He got a heads as well. So he is, in this case, donating a dominant R, and this baby ends up big R, and this baby ends up with a round chin. And again, because I had the expression of chin size, I can also flip for chin, whether this baby's going to have a cleft chin or not. So I scroll down. So we're going to use the letter A. So if it's big A, or big A, little a, our baby's going to have a cleft chin. And if it's little a, that cleft chin will be absent. So I'm going to flip for that. I got heads, so I donate a big A, and you?
You're giving my daughter,
yes,
Flippa, a cleft chin.
Yes, Flippa has a cleft chin. And he got tails, so she still gets a cleft chin. She's heterozygous for a cleft chin. Remember, you could only do cleft chin if you were homozygous dominant or heterozygous for chin size. So this is going to be A. All donated a little a. And the baby is big A, little a. And the baby has a cleft chin. Awesome. Okay, so again,
I don't know if that's so awesome.
I think it's awesome because remember, this is the ugliest baby contest, not a most beautiful baby contest.
Well, not that cleft chins make someone ugly. It's just you don't normally think of a girl having cleft chins. It's more of a male trait, isn't it?
Flippa does. Flippa has a cleft
Okay.
chin. So what we want to do is start, drawing this baby. So round face, prominent chin, round chin, cleft chin. Now I've also highlighted the next trait. This one's skin color. This one is a special case as well. It's not epistasis. So remember, epistasis is where one trait, the expression of one trait controls the expression of other traits. Skin color is what we call a polygenic trait. That means that the expression of a particular phenotype, in this case skin color, is dependent on a whole bunch of other genes. So it takes multiple genes to make a particular phenotype. So if we scroll down, let's take an example of this, or see what we're talking about. So notice I highlighted this in green just so you can pay attention to it. And you'll notice that it says, to determine skin color, assume there are 3 genes involved. Now in reality, there's probably a lot more than 3 genes affecting skin color, plus the environment. But in this case, let's assume 3 genes located at 3 different loci, which means 3 different locations on a chromosome, and that these are going to work together to produce a particular skin color. So we're going to call the first set of genes A. Okay, so it'll be an A locus. Remember, locus is just a location on a chromosome. B will be the second gene, and C is the third gene. And so what we're going to have to do for skin color is we actually have to flip 3 times, one for each of these 3 genes. So I'm going to flip to find out, do I donate a dominant A or a recessive A for the first gene? Then I'm going to flip again, do I donate a dominant B or recessive B for the second gene? And then same thing for the C. So, and then he's going to do the same thing. So for the A locus, I got heads, so I donated a dominant A. For the B locus, I got heads, so I donated a dominant B. And for the C locus, I got tails, so I donated a recessive C. So I got capital A, capital B, lowercase c. You do 3 times now. So now he has to figure out what he's donating.
Do you have your thing memorized?
Yes, I do. Okay, so he got tails, so he's lowercase a. Got tails again, lowercase b. And lowercase c. So he's recessive for all 3 loci. I was dominant for the first 2 loci and recessive for the second. So now you come down to the skin color model.
Uncanny.
Yeah.
How you're always getting the heads, and I'm always getting these tails.
I'm always ahead. Anyway, head of the household. Okay, so so now you got to come down to this chart and see. So if you got 6 dominant alleles, the baby's going to be very dark black for skin color. 5 dominant alleles, very dark brown. 4 dark brown. 3 medium brown. 2 light brown. 1 dominant allele is ivory, and no dominant alleles is very fair. So I'm just going to write here what we got here. So mom, that's me. I'm going to put this in red so we know that this is just what I'm writing in here. So mom, I got eggs that carry big A, big B, and little c. And dad, he got recessive for each. So little a, little b, little c. Well, you put us together. We have big A, little a, and then we have big B, and little c, little c. So in total, we have only 2 dominant alleles out of these 6. And so I'd go down here, I'd say 2 dominant alleles. That means we have a light brown baby. Okay, so we have a light brown baby. So we're going to come up here.
That'd be, I guess that'd be.
That works.
Kind of light brown.
Okay, so for here, for the alleles that mom donated, I'm going to go big A, big B, little c, because that's what I flipped for. Dad got little a, little b, little c. The baby's genotype, and remember, you want to, by convention, keep the same letters together because these represent alleles for the same trait. So big A from me, little a from dad, big B from me, little b from dad, and little c from me, and little c from dad. And this baby, by that chart, was light brown skin. Okay. The rest of the traits are pretty self-explanatory. Hair type is a case of incomplete dominance. So that's where instead of the dominant allele completely masking the recessive allele, it's incompletely dominant, so they blend. So if you come down to hair type here, you'll see, because it's a case of incomplete dominance, if you're homozygous dominant, you end up with very curly hair. If you're homozygous recessive, you end up with straight hair. But if you are heterozygous, you end up something in between, wavy hair. Okay, so that's a case of incomplete dominance.
That's pretty cool.
Yeah, and etc. You know, eye color is one of these really strange, that's another polygenic trait. And in fact, in mice, they've found that there's at least 60 genes involved with producing eye color. So, you know, this is a good example is our real daughter, Evangeline, has very unusual eye color that's kind of turquoise and green and blue all mixed. And our eyes are totally different from that. And there's a lot of genes involved. So it's very hard in reality to predict what color eyes a baby's going to end up with. But anyway, you'll just go through all of these and you'll end up with a phenotype for each of these. And based on that, take out your colored pencils and draw your ugly baby. Or if you prefer, you can open up a new document in Word, and you could also do this in PowerPoint, for example. And you can insert shapes and color them and all this kind of stuff. This little squiggly line right here will actually allow you to draw. So you can draw your baby. So anyway, have fun with that. But if you want to just go the old-fashioned way with colored pencils, just be sure to take a digital photograph of your ugly baby, maybe with your camera. And there's a place in here when you go down to the follow-up questions, which are right here. So answer these follow-up questions. Think about the different kinds of inheritance patterns we talked about. And in here, paste that JPEG picture of your ugly baby. And then post the picture also on the discussion board for this lab, and we will vote on the ugliest baby. So we'll have an ugliest baby contest, and the winner will get a prize. Finally, second part of the lab is just practicing genetics. So these are pretty self-explanatory. If you have any questions, certainly ask me on the discussion board. This first one, you're assuming that both parents are heterozygous for 6 fingers. So they both have 6 fingers. In this case, 6 fingers, big F, is dominant to 5 fingers, little f. And you fill out the chart. So phenotype, where it says we're both 6 fingers. So dad is 6, mom is 6. Genotype, well, it says we're both heterozygous. So the dad would be big F, little f. Mom would be big F, little f. Here, it wants the individual gametes. So just like we did before, if dad is big F, little f, what kind of gametes could he make? He'd be big F or little f. And same thing for mom. And then based on that, figure out the probability of having a 6-finger child or a 5-finger child. And you'll put those percents up there. And then basically, the rest of it's all genetics problems. And I've already put in here for you some Punnett squares to help you along. So you can just fill those out. Some of these are more logic problems. They actually, instead of having, like, this is a dihybrid cross, so you'll have to fill that out. But some of them, like towards the end, are really just logic problems. You just got to think about them. But anyway, I encourage you to participate on the discussion board with your colleagues on how to fill these out. They're fun. There's lots of resources on the web. And of course, I'm always here to help. Thanks so much. And I look forward to hearing how your lab's going. Bye.